This is Drew, and I'm going to give you an audio enhanced presentation titled Pain Sensitization for your MM1 class. So let's begin. At the end of this AEP, you should be able to discuss mechanisms involved with peripheral nociception, and then we want you to think about PT intervention strategies to go along with this mechanism, as well as identify how central sensitization might develop in a subset of your patient population, again, considering PT intervention to work with this patient population. So here's how we're going to do this. We're going to begin with a brief review of normal pain processing and talk about what normally happens after injury. Then we're going to move on to pathology. So what would a pathological peripheral sensitization situation look like, and then how central sensitization might develop in a subset of your population. So let's begin by reviewing classes of nociceptors. So this should be familiar to you, right? We have different types of nociceptors that transduce different types of sensation. Thermal sensation, mechanical sensation, and polymodal sensation. So your A delta fibers and your C fibers will transmit nociceptive information to your spinal cord and eventually to your brain. And then we shouldn't forget about silent nociceptors. So normally these neurons are not firing, okay? But ongoing inflammation and chemical agents will reduce their firing threshold, therefore allowing them to become active. And let's talk here about C fibers and A delta fibers. As we discussed in neuroscience this last semester, A delta fibers are responsible for giving you that first pain that you might feel, let's say, if you hit your thumb with a hammer. Whereas as time goes on, you'll feel second pain coming from C fibers, which is that throbbing, deep pain. Again, first pain carried by the faster A delta fibers, second pain carried by the slower unmyelinated C fibers. And so here's a review of the typical pathway of the lateral spinothalamic tract, which transmits pain to your brain, right? So we have nociceptors coming from the periphery, and they would be out here, and they transmit information through the dorsal horn to the dorsal horn of the spinal cord, right? We remember from neuroscience that then this pathway crosses and ascends in the lateral spinothalamic tract, synapsing at the thalamus, and then passing its information on to cortex here, where your patient will then consciously feel pain. Again, here is a diagram, or I should say, this is a diagram of the DCML pathway, okay? So not the pain pathway, but as we recall, the light touch pathway. And this is what happens normally. The DCML comes in, ascends ipsilaterally here, and doesn't cross up until the level of the medulla, where it hits the thalamus and then cortex. Notice this area here, though. This is where a collateral is given off into the dorsal horn of the spinal cord. Now this is going to be important later on in the lecture, especially when we talk about mechanisms to reduce pain that we feel. So the dorsal horn, here's how it's organized. And what I want to kind of touch on is that you can think about the more dorsal areas of the dorsal horn here as being areas that receive pain information or nociceptive information, whereas the more lower areas, ventral areas of that dorsal horn receive more non-painful mechanosensory information. This will all come into play later in the lecture. And so, a review of neurotransmitters. Glutamate is responsible for transmitting fast potentials in the dorsal horn, whereas neuropeptides, for instance, substance P, they are responsible for this slow excitatory postsynaptic potentials in the dorsal horn, And what's important about neuropeptides is there's not a specific mechanisms to reuptake that neurotransmitter. Therefore, it can kind of diffuse away from that site and might be responsible for more diffuse pain and pain that's perceived by patients that's not localizable. Just a fun little neuroscience trivia fact, nociception is the only human sense that becomes more sensitive with repeated exposure. So if we think about after we exit a movie theater, our eyes are not quite adjusted to the brightness of the outside light, but then we quickly become adjusted. Same thing with the auditory system and white noise. We soon learn to ignore it. Olfactory system, same deal. But nociception is the only sense that becomes more sensitive with repeated exposure. My example of this is I had a tattoo, and I was assuming that I would get used to the sensation of the tattoo, and I feel like getting the lining of the tattoo is no big deal, but as if you shade in or color in at a certain area over and over and over again, the same area, you become sensitized to that stimulus. So, what normally happens after injury? 5 main chemicals of inflammation come into play. So we'll first start with histamine. So histamine is released from mast cells that are damaged in response to tissue injury. Histamine directly activates C fibers. Bradykinin directly activates both A delta and C fibers and increases prostaglandin production. Prostaglandins are generated during inflammation and are responsible for sensitizing nociceptors. Cytokines come from these macrophages that release interleukin-1 and tumor necrosis factor. Okay, these two, this system here will sensitize nociceptive terminals. And then neuropeptides such as substance P will cause edema and histamine release. I made the diagram like this to show that these chemicals can either work alone or they can all activate each other and work in tandem, all to sensitize nociceptors. So, typically, here's what happens. After the injury, these chemicals are released, leading to edema and inflammation, and then the ongoing presence of edema and inflammation will lead to sensitization. In normal circumstances, neuropeptides such as substance P are released here in the periphery, as well as centrally here in the dorsal horn. And here's a visual representation of the previous slide. So we have the C fiber out in the periphery. Substance P is released in response to tissue injury into the dorsal horn, leading to nociception. But it's important to know that in normal circumstances, there is no production of substance P by A beta fibers. Remember, A beta fibers are responsible for transmitting light touch information. Light touch information. So here's our main takeaway points. What normally happens after injury? Well, inflammation happens, chemicals from various cells are released, and nociceptors get sensitized. We discussed the 5 main players of inflammation, and normally only C fibers are producing that substance P in the dorsal horn. So, how about some treatment ideas that can come along with this? The first 2 ideas are very passive treatments. Okay, here's how they might work to reduce pain. But I want you to know that these first 2 interventions, and Dr. Albin wants to reaffirm that these are only short-term treatments and they're not shown to have long-term efficacy. These are likely inappropriate for folks with chronic pain. So cold or ice can reduce inflammation and decrease nerve conduction velocity to reduce pain. Hot packs are known to increase circulation and also sedate those nerve endings. Now the next two are more active interventions, especially regarding patients with chronic pain. So let's talk about tactile input. Tactile input can promote gating of pain by increasing non-painful input. An example of this would be a therapist giving gentle pressure. And then of course we have motion. Active motion and tactile motion can cause a stimulus to be sent through this mechanoreceptor fiber here in order to gate the pain at the dorsal horn. Also, passive range of motion and active range of motion, they aid in circulation and uptake of those chemical irritants, those 5 players of inflammation. So, in this case, all of these treatment ideas are going to be addressing pain from the periphery. So, we've talked about what normally happens. Now we're going to move on to how ongoing inflammation and edema can lead to pathological peripheral sensitization. Here are 4 examples that we'll go through. Increased synthesis of those TRP channels, which are the channels at the ends of free nerve endings. Activation of silent nociceptors. Increasing the receptive fields for hyperalgesia. And a phenotype switch in A beta fibers. Let's go through each one. So, inflammation, ongoing inflammation, can lead to gene expression in the dorsal root ganglion. What does this mean? This means that the dorsal root ganglion starts to crank out more channels, meaning TRP channels, that then get inserted at the ends of these nociceptors. What does that ultimately mean? That means that a stimulus that would have normally activated a few channels here at the end will activate more, therefore leading to increased peripheral sensitization. How about the second mechanism, activation of silent nociceptors? So ongoing inflammation will activate these silent nociceptors by reducing their thresholds for firing. And now you will receive hyperalgesia and peripheral sensitization. So remember, silent nociceptors are not usually activated by any stimuli, but ongoing inflammation can cause these particular silent nociceptors to become active. What about increasing receptive fields for hyperalgesia? Here's a classic example of a study where they took 8 human volunteers and the researchers applied a mechanical stimulus. And they wanted to see how much can I push on the palm of your hand until you start to perceive it as painful. And they recorded it here in the gray bars. Okay? Then they applied a heat stimulus for 30 seconds right here in the dark red area, which then would cause a sensitized stimulation to that palm. Okay, then they recorded the mechanical stimulus again and said, okay, how much pressure can I push before you perceive it as painful? So first off, they realized that even if they applied the stimulus outside of the area that was heated, it would still be perceived as painful, and that even if they applied the stimulus just a little bit, right, so the pain threshold was just applied slightly, they started to feel pain right away, meaning that those nociceptors here in that area had become sensitized and feel pain quickly. Okay, now here's a figure showing the mechanisms for that burn study that I just presented. So ongoing inflammation leads to a cycle of neuropeptides being produced by nociceptors. This encourages histamine to travel and recruit prostaglandin, bradykinin, and cytokines. thereby spreading away from the site that was initially damaged, away from that site that was initially burned. So notice how all of these factors are working together to cause nociceptor sensitization. And finally, the fourth mechanism behind pathological peripheral sensitization is what can happen is ongoing inflammation can cause a phenotype switch. What does that mean? It means that some A beta fibers that normally transmit nice light touch pain will now start to express substance P. And remember that substance P, if it's dumped on the dorsal horn, will cause that diffuse feeling of pain. So A beta fibers are now able to contribute to the inflammatory hypersensitivity by enhancing substance P transmission in the dorsal horn and exaggerating those responses to stimuli that's formally soft and doesn't feel painful. So here's kind of a review of what we just talked about. Ongoing inflammation can cause pathological peripheral sensitization. How? Increased synthesis of those TRP channels that are found at the ends of those free nerve endings. Activation of silent nociceptors, meaning those nociceptors that are not normally active at all. Increasing the receptive fields, meaning that the painful areas can be felt outside of the areas that were damaged in the body. And finally, a phenotype switch in which those A beta fibers that normally transmit nice, soft, light touch information now will be perceived as painful. Okay, I know that's a lot of content, but let's push forward, onward to central sensitization. Central sensitization, what do we mean by this? We mean that stuff happens in the central nervous system, in the spinal cord, the brainstem, and the brain, that causes enhanced pain transmission. Okay, let's talk about the first mechanism, wind-up. You might have heard this term wind-up. Maybe you've heard of it as dorsal horn wind-up. What happens here? Persistent injury causes pain fibers to keep glutamate around the dorsal horn, glutamate. Okay? By glutamate being persisting in the dorsal horn, normally only AMPA receptors are active. An increased presence of glutamate causes NMDA receptors to also become active, and then, therefore, the dorsal horn becomes hyperexcitable. You might recall this mechanism from neuroscience. We discussed this as long-term potentiation. Okay, another mechanism for dorsal horn central sensitization is, following persistent injury, those A beta fibers can actually project up into the dorsal parts of the dorsal horn where normally nociceptors are synapsing. Okay, we'll discuss these two mechanisms. So, what is wind-up? Persistent injury causes C fibers to keep firing. Okay? So therefore there's more glutamate located around the dorsal horn. This allows for NMDA receptors to also become active, leading to improved transmission of pain signals upwards to the brain. We call this dorsal horn windup or dorsal horn hyperexcitability. Now for the second mechanism of dorsal horn central sensitization. Remember, this is how the dorsal horn lamina are normally organized, right? Pain is normally received up here. Mechanosensation is generally received down here. Now let's go through this next slide. It's a little complicated, but I think we're going to get through it. So here's the light touch system in blue here. And we're going to depict this as a feather. The nociceptive fibers are here in black, okay? And the painful stimulus is going to be depicted as a fire. Now let's zoom into the dorsal horn. By zooming in here, we can see this is what normal looks like, right? A beta fibers synapse down, C fibers synapse up. But multiple animal studies have shown in the chronic pain state, the light touch fibers encroach into the C fibers lamina here. What does that mean functionally? That means now light touch will be perceived as painful. So here's a quote from one of these papers down here. The author's name is Licken et al., 1996. The authors say, large A beta fibers sprout into lamina 2 where small C fibers are found. This may explain the intractable touch-evoked pain that can follow injury. Main points of central sensitization. Remember, dorsal horn windup. This is another example of long-term potentiation that can happen at the dorsal horn. And number 2, A beta fibers have been seen to project into nociceptors lamina. I also wanted to put this slide in that central sensitization can also refer to changes in the brain. Changes do occur in higher levels such as the cortex. We won't discuss these mechanisms in this particular AEP, but be aware that this does indeed occur. So how would we approach treating patients with central sensitization? Here's a nice quote from a 2013 PT Journal article. It's looking at osteoarthritis and central sensitization. And I love the quote, chronic pain in people with OA is related more to neuroplastic changes in the nervous system than to a local condition of the joint. So, I propose a PT intervention of course you're going to treat the individual and the individual's impairments, but then you can also use strategies that these authors proposed. Pacing, time-contingent graded exercise, and neuroscience education. So let's talk about activity pacing. You can use pacing to lower pain flare-ups. The big picture to this is just because you feel good one day doesn't mean you're going to go and run a marathon. Okay, but also if you feel in pain the next day, that also means you should probably still get up and move at least a little bit. Now, a time-contingent approach means the patient doesn't simply cease to exercise once local pain severity increases. Instead, the patient adheres to a predetermined exercise intensity and duration, and we work to help the patient interpret this pain and the pain increases as non-threatening, which leads into neuroscience education. The aim for neuroscience education is to reconceptualize chronic pain in a way that it is no longer regarded as threatening. The goal is that chronic pain will no longer be understood as tissue damage, but simply noise in the sensory system. So, let's talk about mechanisms for how exercise can reduce pain. Exercise releases beta endorphins from the pituitary gland and the hypothalamus. This allows for these feel-good sensations by activating opioid receptors. So the hypothalamus, through its projections on the periaqueductal gray, has the capacity to activate descending nociceptive inhibitory mechanisms. So one animal study showed that both regular exercise, kind of this submaximal rhythmic exercise, as well as just one single bout of exercise, both of these conditions increased opioid receptors in the brain. And we can assume that this happens in humans as well. So here's a figure showing that, of course, we have the ascending pain systems, but then, once we start exercising, exercise activates these descending pain modulation systems, which will go eventually to inhibit the pain signals at the level of the dorsal horn here. So, a few treatment ideas for our patients with central sensitization. Of course, we can treat the individual impairment-based approach. We can deliver time-contingent graded exercise, okay, knowing that exercise is going to activate those descending systems to help with pain control. Activity pacing. We can use pacing to avoid pain flare-ups. Again, just because you feel good one day doesn't mean you should go run a marathon. But if you feel in pain the next day, you should still get up and move. Neuroscience education. We're aiming to reconceptualize chronic pain in a way that it is no longer regarded as threatening. Of course, we can always recruit other disciplines to help us out. Medicine, possible pharmacology, and counseling. We know that cognitive behavioral therapy has been shown to be effective. In all of these cases here, all of these treatment ideas are now addressing pain at the central nervous system level. All right, so summary of our AEP. We went through peripheral nociception. What happens with this nociceptor activation and inflammation? We're thinking about interventions using tactile input, pain-free motion, treating at the periphery. Whereas central sensitization and the mechanisms we just discussed, we're proposing treating individual impairments, grading exercise, pacing, neuroscience education, and therefore treating both at the periphery and at the central nervous system level. And that's all I have.